Next lesson is biotechnology and its applications. In the part of biotechnology, the gene manipulation have been studied, that is a manipulation of the genes where the foreign gene particle is introduced into the native organism. So here this leads to the formation of GMOs, that is genetically modified organisms. If they are crossed, we call them as a GMCs, that is genetically modified crop plants. Before going to this gene manipulation, so let us study something about the process called as a green revolution. The word green revolution was given by the William Gott scientist. So here this concept was first developed by the Norman E. Borlaug, or Norman Borlaug scientist. So here what is the work done by this Norman Borlaug scientist is, he developed an dwarf variety of maize which is having the high yielding ability, and he is considered as a father of green revolution because he was responsible for increase in the production of wheat variety where the plants are green in color. Whereas there is one more scientist called as M.S. Swaminathan. He is an Indian scientist who introduced this dwarf variety of wheat that was produced by Norman E. Borlaug to India, and he done hybridization with that dwarf variety of wheat and produced new varieties of wheat plants in India. So there, because of his work, the yield of maize, yield of this wheat was highly improved in India. So that led to the improvement of yield or the crop production in 1960s. So he is considered as a father of green revolution in India, and he also tried in other plant species or other crop plants like in rice. There also he got succeeded, and he tried in some other commercial varieties of plants also. So this is a way how the green revolution had compensated or balanced the nutrient requirements in 1960s. But as those requirements are not sustainable or not enough in the course of the time period, nowadays it is not enough. So now we are going for the gene revolution. So gene revolution means we are manipulating the sequence of genes, that is called as a gene manipulation process. So because of the gene manipulation, we are producing the genetically modified organisms or genetically modified crop plants. So the plants which are produced by this process are called as transgenic plants, or the organisms are called as transgenic organisms, where the gene sequence have been changed because of the gene revolution, or we can call this as a gene manipulation process. What is the intention of producing this genetically modified crops? So first thing is to get high yielding varieties or to improve the crop yield of the plants. The second one is producing resistance in the plants, or the plants with disease resistant properties are produced by this gene manipulation. And this is a way how this the abiotic stress can be overcome by various varieties of plants, and also disease resistance or disease resistance to various types of viruses, bacteria, fungi, nematodes, or various types of pathogens is possible by this process. And let us take some example here, that is a golden variety of rice have been produced by incorporating genes into type A variety of rice, where it is having high yielding in the vitamin A. So that is golden rice variety. So this is a one which is rich in nutrients, which is rich in vitamin A. And as we know that when vitamin A is there, it can overcome the one of the diseases like a blindness in human beings. Likewise, there are many nutritional improvements, or we can say resistant to various types of environmental stress and strains, sustaining the, or we can say overcoming the biotic and the abiotic stresses, disease resistant properties, resistance to pests. Likewise, various types of new characters are incorporated into the genetically modified crop plants because of this gene manipulation process. And here let us take some of the some other examples. Like we already know that there is a bacterium called as Bacillus thuringiensis. So this is a one which is called as BT. Bacillus thuringiensis. This is an bacterial biopesticide. So what is the importance of this bacterial biopesticide? This bacterial biopesticide, when it is useful for controlling the insects or the pests. How it is helping in the process means, actually these toxins are there, are with the toxins which are present in it are the protoxins which are in the inactive form. When these protoxins, when they enter into the gut of that insect, in the midgut, because of alkaline pH, what happens, the protoxins are converted into the active form. And when these protoxins become converted into active form, what happens is they start their activity on the midgut of the epithelial cells. Gradually it helps in the formation of a perforation leading to swelling and gradually leading to the death of the insects. That is a way here the Bacillus thuringiensis is acting as one of the best biopesticide, that is bacterial biopesticide. In the same way, the viral biopesticides are also there. The best examples are nuclear polyhedrosis virus, which belongs to baculo group of viruses. That is one other example. The next one is here whenever this Bt is there, so they are symbolized by crygenes. And here there are three types of genes are there: Cry1a, Cry2a, 2b and Cry1b. So these are three types of the genes which are specified because of the activity of the insect killing nature. So like these are the one which are helping in controlling of cotton boll worm and the corn borers. So these are the two types of insects which are controlled because of the crygenes. Then here there is one more process is there to control the nematode parasites or nematode pathogens. So it is done by RNA interference. We call that as RNAi. That means I means interference. Actually we know that RNA is the one which will participate in a process called as translation that leads to the formation of a protein molecule. So here in this process we have this RNA interference means the silencing the mRNA molecule by not allowing it to produce the protein molecule. So how it is done means, actually they into this organism they will introduce one more single stranded RNA. This single stranded RNA will go and bind to the RNA which is responsible for the synthesis of the proteins. When this single stranded RNA go and bind to that other single stranded RNA, it forms into a double stranded RNA molecule. When a RNA become double stranded, it does not allow the formation or it does not participate in the formation of the proteins. That is the way how this the introduced single stranded RNA will go and interfere the, or we can say that inhibit the process of the translation of the RNA molecule. That is the way how the nematode parasites are controlled by RNA interference process. Then the next one is related to the production of insulin. We know that insulin is the one which will control the sugar levels in blood and urine in human beings. So here it is produced, actually it is produced by a company called Eli Lilly company, and it's an American based company. This had produced two chains of insulin. One is chain A and chain B. They artificially synthesized these two chains, and they, after synthesizing them, they introduce these two chains into Escherichia coli. So what is happening in the Escherichia coli means, when they enter into Escherichia coli, separately they will multiply their number and they get multiple copies of them. Then afterwards, between this chain A and chain B, so the disulfide bonds are formed. So when disulfide bonds are formed between chain A and chain B, it leads to the production of insulin. So that is a way how the insulin production is done with the help of Escherichia coli, and that insulin which is produced with the help of this Escherichia coli is called as humulin. So this is a way how the production of insulin can be have been enhanced by using this artificial method that is done by a company called as Eli Lilly company, which is an American based company. So likewise, this is one other aspect of application of genetic engineering or biotechnology. So likewise many other applications are there, like production of the disease resistant varieties. For example, papaya, the resistance to ring spot disease which is caused by viruses. In the same way, cotton, the resistance to insects, that is BT cotton we can say. In the same way, if you take a roundup variety of soya bean which is resistant to nematodes. Likewise, various examples are there where they are having the resistant property. Like in potato, resistance to fungi called as Phytophthora. The same way, tomato, resistant to the bacteria called as Pseudomonas. And when it is coming to the part of a flavor saver variety of tomato which is having the bruise resistant character, the flavor saver variety of tomato which is having a bruise resistant property. So likewise this is one other character. And the next one is, we know that basmati varieties of rice is there. So basmati variety of rice, so this is a one which is having ability of resistance against biotic and abiotic stresses. Okay. Then coming to next one, that is in brassica, production of male sterile plants. So what is the importance of producing male sterile plants in brassica? We know that during artificial hybridization process, emasculation is done in the bisexual flower of female parent. So emasculation means the removal of the anthers or stamens. So here, if the plants are male sterile, we can use the male sterile plants directly as a female parent. The flowers are used as a female parent directly without performing emasculation process. So such type of plants are produced by this biotechnology. Likewise, if we take many applications are there. Then let us come to some of the techniques where molecular diagnosis is important here. So related to the molecular diagnosis. We know that the disease is identified by going through some diagnosis tests. So here the diagnosis or test will rectify the presence of, it will identify the presence of various pathogens in the substances or the blood or somewhere, whatever it have been tested. But here early diagnosis is so tough, it is not an easy job to be done. But here there are some techniques called PCR technique and ELISA technique. PCR means polymerase chain reaction, ELISA means enzyme linked immunosorbent assay. So these are the techniques which are helping in early diagnosis of the diseases. So what they will do means they will amplify the genes which are responsible for the disease and they will identify the disease. So likewise these are having some important role in the early diagnosis of the diseases. And generally ELISA is based on the antigen antibody property. So it is identified, okay, it will done by identifying the number of antigens, or it will identify when the number of antibodies are more against any antigen. So likewise it is based on the antigen antibody principle. So likewise these are some of the examples related to the molecular diagnosis where PCR and ELISA are having some important role. Then so whatever we are saying that because of biotechnology, the new plants or gene manipulation is done where the genetically modified crops and genetically modified organisms are produced. But up to which level they are safe to use or to consume or to use them for various purposes. So that is concerned with the biosafety issue. That is, there are many doubts related with the plants which are produced by this biotechnological process. They have some doubts, that farmers particularly, they have some doubts that is it safe to use such type of grow such type of plants in the environment, because if such type of plants are grown, will it lead to any pollution of the environment, or will it change the normal atmosphere or nature which is present around the plants? The second one is, if we consume those biotechnologically modified plants, so will it causes any allergies in the human body or allergies in the animals, whoever will consume those genetically modified crops? And here what is the natural gene constituent which is present, that is unknown after some years. And there is a chance of production of super weeds also. It is very tough to eradicate those super weeds which have been produced by this biotechnology process. Likewise, there are many issues related to up to which level it is safe to use this genetically modified crops or genetically modified organisms. So for that purpose, there are many organizations which had come forward for explaining that biosafety issues. In that one, the most important one is a GEAC, Genetic Engineering Approval Committee. GEAC. This is mainly working on the aspect of the how up to which level the genetically modified plants are useful for human beings and useful for the nature and also for the all organisms whoever all surviving on the earth. So that is about the biosafety issues. And coming to the biopiracy. So we know that there are some countries who are using some native plants or the native organisms of some other countries without giving information to that native country. So likewise, one example is there where in Basmati is a native of India. So here there were 2 lakhs varieties of Basmati varieties, but nowadays 27 documented varieties of Basmati are there. And these Basmati variety are known for the aroma and taste. And American companies or American country, what the American government had done is they had taken the Basmati varieties and crossed with some other varieties and produced a new variety of Basmati, and they are saying that this is a native to their country and they had taken patent on that Basmati variety. So this is a way how without taking the permission of the native country, they are utilizing the product or the plant species which is native to that particular country. That is what we call it as a biopiracy. So here our government have to take some steps, or our government should be in a pioneering way where we have to take the patent of native crops like turmeric, which is one of the most important medicinal plant. Okay, these are the various issues related to the biotechnology and its applications. So now let us come to the question part where some important questions, or we can say exam point of view, expected questions, let us discuss in the next part of this biotechnology and its applications. Question number 1. Who among the following is instrumental for the success of green revolution in our country? Options: Norman Borlaug, 2. M.S. Swaminathan, 3. Herbert Boyer, 4. William Goud. Okay, here let us take the first scientist. Already we said that Norman E. Borlaug is a scientist who produced that dwarf variety of wheat, which is a high yielding variety. He is called as the father of green revolution, but he is not concerned to India. But when it is coming to Swaminathan, already we had discussed in the introductory part where he is a person who introduced the dwarf variety of wheat to India, which was produced by Norman Borlaug, and he done hybridization experiments with the dwarf variety and produced a new variety of wheat, and he was successful in increasing the yield of wheat many times to the normal yield that was at that particular time. So it was in 1960s where there is a great increase in the production of wheat, so we call it as a green revolution, and Swaminathan is called as a father of green revolution in India. So question number 1, the correct answer is 2. Question number 2, identify the correct statement from the following with reference to the genetically modified organisms, GMO. These are: first option, organisms whose genes have been altered by manipulation. 2. Organisms whose chromosomes have been altered by manipulation. 3. Organisms whose cytoplasm has been altered by manipulation. 4. Organisms with change in some base pairs through selection. Okay. Here in the first option, already we know that genetically modified organisms means where the gene manipulation is done. Gene manipulation means where the new genes are introduced into the organism so that the native sequence, whatever the original sequence of genes are present in the organism have been changed. So that means when the manipulation is done by altering the gene sequence, altering of the gene sequence, so we get an organism called as a genetically modified organism. So question number 2, the correct answer is 1. Question number 3. Which of the following is a pest resistant crop developed through genetic engineering technique? Options: 1. BT cotton, 2. BT rice, 3. BT groundnut, 4. BT sugarcane. Okay, in all the options it is BT is common, that is Bacillus thuringiensis. This is a bacteria which is having the biopesticide, which is used as biopesticide. So for killing the insects, it is a source of living organism. So from that we can say, from all the options we can say that the one which is used as a pest resistant crop, that is a BT cotton variety. BT cotton variety. So question number 3, the correct answer is 1. Question number 4. The process of preventing the translation of mRNA is called as. Options: 1. Transcription, 2. Transduction, 3. Transformation, 4. Silencing. Okay, here already we had discussed that RNAi, that is RNA interference, where a single stranded RNA is introduced into the organism. That single stranded RNA will go and bind to the mRNA molecule. When it is going and binding to the mRNA molecule, that mRNA molecule will become a double stranded RNA. So here when a double stranded RNA is there, the translation is prevented. This process of preventing the translation is called as silencing process. Silencing process. But in the options it is given as transcription, transduction, transformation. These three are nowhere related to the silence, or we can say preventing the translation of mRNA molecule. So question number 4, the correct answer is 4. Question number 5. One of the application of biotechnology is the production of human insulin. It has been now produced from one of the following. Options: 1. Plants, 2. Viruses, 3. Animals, and 4. Bacteria. Okay, here human insulin, which is an hormone which will control the sugar level in human body, and here it is produced by using an bacteria called as Escherichia coli, where already we discussed that Eli Lilly company of America, so that had produced two artificial chains, chain A and chain B, which have been introduced into this bacteria, where that help in the formation of insulin. So that type of insulin is called as humulin. So question number 5, the correct answer is 4. Question number 6. Structurally, the human insulin is composed of one of the following. Options: chains A and B, chain A and C, chain B and C, and fourth one, chain B and D. So here human insulin contains two chains. One is chain A and chain B. These two chains are binded with disulfide bonds, which help in the production of human insulin, that is called as humulin, by using this bacteria called as Escherichia coli. So question number 6, the correct answer is 1. Question number 7. Cryprotein is obtained from one of the following. Options: 1. Viruses, 2. Bacteria, 3. Bacteriophage, and 4. Fungi. Okay, here this is related to a bacteria called as Bacillus thuringiensis, that is what we call it as a BT, and which is used as a biopesticide. And this one is having inactive proteins called as protoxins, toxins called as protoxins, and these are in the crystal form, and these are specified by a genes called as crygenes. And as we said that Bacillus thuringiensis is a bacteria, so that cryproteins are obtained from bacteria. Question number 7, the correct answer is 2. Question number 8. Name the genus of type A transgenic variety. Options: 1. Solanum, 2. Triticum, 3. Brassica, 4. Oryza. Okay, here it is related to the type A transgenic variety. So here type A is a variety into which the genes have been introduced, which had produced a new variety of plants called as golden rice variety. So it is having vitamin A. That means it is rich in vitamin A. So here, as we are saying, it's a rice plant. The genus to which this type E transgenic variety belongs to is Oryza. Oryza. So question number 8, the correct answer is 4. Question number 9. Study the following table. Here column 1 and column 2 are given. And in this first one, disulfide bonds, human insulin. 2. Molecular diagnosis, gene therapy. 3. Cryproteins kills certain insects. 4. PCR, polymerase chain reaction, detect variations in genes. The correct combination is options 1, 1 and 2, 2, 2 and 4, 3, 1 and 3, 4, 1 and 4. So let us come to the first and second columns here. 1. Disulfide bonds. So already we said that these are the disulfide bonds are present between the chain A and B, which are responsible for the production of human insulin. So this column is correct. That is, the combination is correct. First one is correct. Then coming to molecular diagnosis. So here molecular diagnosis is related to the early diagnosis of the diseases. The molecular diagnosis, that is early diagnosis of diseases, that is done by PCR technique and ELISA technique. So it is not related to gene therapy, so that is incorrect. Then coming to cryprotein. So here cryproteins are the proteins which are the part of the toxic materials produced by the Bacillus thuringiensis, or it is used as a biopesticide, so it will kill pests or kill certain insects. So this statement is this combination is correct. Then the fourth one is PCR, polymerase chain reaction, detect variations in genes. So it is not related to detection of variations in genes, but it is useful for the molecular diagnosis, or we can say that early diagnosis of the diseases. So from these options we can say that first one combination is correct and third combination is correct. So question number 9, the correct answer is 3. Question number 10. Identify the correct statement with reference to the process of RNA interference, RNAi, from the following. Options. 1. Silencing of specific mRNA by a complementary RNA molecule. 2. Silencing of specific complementary RNA molecule by a sRNA molecule. 3. Silencing of specific mRNA molecule by a rDNA molecule. 4. Silencing of specific mRNA molecule with a plasmid vector. Okay, here the question is related to RNA interference, that is RNAi. So what is done during the RNA interference? That is the translation of mRNA is prevented. So mRNA translation is prevented by a process. Translation is prevented, and it is done by introducing a single stranded complementary RNA into that organism, so that that complementary RNA go and bind to this mRNA, and this mRNA now converted into double stranded RNA molecule. When double stranded RNA is formed, it does not help in the translation process. So that silencing of mRNA, specific mRNA by a complementary RNA is called as RNA interference. So question number 10, the correct answer is 1. Question number 11. tDNA is associated with one of the following. Options. 1. Retroviruses. 2. Bacteriophage. 3. Bacterium. 4. Fungi. Okay, here the question is related to tDNA molecule. tDNA is the part of the Ti plasmid, which is present in a bacterium called as Agrobacterium tumefaciens. And already we have studied in the earlier part of our lessons that Ti plasmid is a natural plasmid, or natural vector which is used in the genetic engineering process for transfer of desirable genes from one organism to the other organism. But as we know that it's a tumor inducing plasmid. So this ability of tumor inducing, it's a pathogenic nature. This can be prevented by a process called as disarming process. Then we can use this TA plasmid for the genetic engineering process where it is used as a vector. So tDNA are present in Agrobacterium tumefaciens, which is an bacterium. So question number 11, the correct answer is 3. Question number 12, identify the correct statement with reference to these term biopiracy. First option, use of plants to recycle environmental pollutants. 2, it is a technique used to identify criminals. 3, use of bioresources by multinational companies without proper authorization from countries concerned. 4, fear of transferring toxins to human from genetically engineered crops. Okay, here biopiracy. So already we had discussed that biopiracy is a process where without prior authorization of the concerned country, if any multinational company is using the products or the plants which are belonging to that particular country, so that is called as a biopiracy. That is exploiting the bioresources of that particular country without giving any specific information. So that we can say the use of bioresources by multinational companies without proper authorization from countries concerned. So question number 12, the correct answer is 3. Question number 13, which of the following crop variety was originally grown by Indian farmers but patent rights for it were obtained by an American company? Options: 1, golden rice, 2, basmati variety, 3, flavor saver tomato, 4, BT cotton. So here first one, that is given as a golden rice variety. So we know that golden variety of rice is the one which is rich in vitamin A. It is not related to the obtaining that is biopiracy process. Then let us come to the basmati variety. So basmati variety is also a rice variety which have been used by this American company, so that without the authorization of Indian, because basmati is a native to India. Then third one is a flavor saver variety of tomato. So flavor saver variety of tomato is the one which is having the bruise resistant property. Then fourth one is a BT cotton, which is having the resistant to pests, or it is used as a BT is used as a biopesticide for controlling the insects. So out of this basmati variety is the one which is used by multinational companies of America without authorization of the native country, that is India. So question number 13, the correct answer is 2. Question number 14, under what conditions the inactive protoxins of the bacterium turn into an active form of toxin? Options: 1, acidic pH of the gut, 2, alkaline pH of the gut, 3, neutral pH of the gut, and 4, concentration of enzymes in the gut. So here this question is related to the BT toxins which are produced by the Bacillus thuringiensis. So here when this Bacillus thuringiensis which are in the inactive form, we call them as a protoxins which are in the inactive form. But when they enter into the gut of that is a midgut of the insect, what happens is they, because of the alkaline pH of the midgut, what happens, these protoxins become activated and they start converting into active toxin, and this active toxin will act on the midgut of the epithelium tissue, gradually leading to swelling of the tissue and killing the entire insect. So at which pH they are becoming active means at alkaline pH, which is present with alkaline pH of the gut. So question number 14, the correct answer is 2. Question number 15, the function of GEAC is options: 1, to ensure the safety introducing GM organisms, 2, to develop the technique of genetic engineering. 3. To develop super weeds. 4. To create an adverse impact on the environment. So here actually it is related to the biosafety mechanism. Already we said that GEAC, that is Genetic Engineering Approval Committee, will be there. It is concerned with the safety or biosafety of the living organisms. That is, we know that genetically modified organisms are there, GMOs, including the crops. That is production of transgenic animals and also transgenic plants. Here up to which level these GMOs are safe to introduce into the field, and also the usage of or utilization of these transgenic organisms for various purposes, so that it is concerned to ensure the safety of introducing genetically modified organisms. So question number 15, the correct answer is 1. Question number 16. What modification is done on the TI plasmid of agrobacterium to convert into a cloning vector for genetic engineering purposes? Options: 1. Several new genes have been added into it. 2. It has been converted into non-pathogenic form. 3. Its molecular weight was increased. 4. Several markers have been introduced into it. Okay, here this is related to the TI plasmid which is present in the Agrobacterium tumefaciens bacteria. So here we know that it is having the ability called as tumor inducing property. It is a pathogenic in its nature, but at the same point of time this is because of tDNA, but here this can be used as a vector in the genetic engineering by a process called as a disarming process. Disarming process. During the disarming process, what they will do means they will convert this pathogenic form into non-pathogenic form. So that means when we have to use this as a vector in the genetic engineering, the TI plasmid is converted into a non-pathogenic form. Question number 16, the correct answer is 2. Question number 17. Study the following lists. List 1, transgenic plant. A. Papaya. 2. BT cotton. C. Roundup Ready soybean. D. Tomato. List 2, resistant to. Options: 1. Herbicides. 2. Ring spot virus. 3. Phytophthora. 4. Insects. 5. Pseudomonas. The correct match is first option A2, B5, C1, D4. Second option A2, B3, C4, D1. Third option A3, B1, C5, D2. Fourth option A2, B4, C1 and D5. So let us come to the plants, transgenic plants which are given in list 1. First one is a papaya plant. Here papaya plant is one which is having that is transgenic papaya plant is having the resistance against the ring spot viruses. Resistance against ring spot viruses. Ring spot virus have been given in option number 2. Then B is the BT cotton, and we know that BT cotton is a biopesticide, or we can say it is having the resistant against the insects. So that Roman number 4, that is resistance against the insects. Then C is Roundup Ready variety of soybean. Roundup Ready soybean. So here this is a one which is having resistance to herbicides. That is Roman number 1, herbicides. Then D, tomato. Actually when it is a tomato, there are two characters have to be considered. One is resistance against Pseudomonas is one, and second one is the transgenic tomato variety called as flavor saver variety, which is having the bruise resistant property. So out of these two, which one is given here? It is a Pseudomonas, that is Roman number 5, Pseudomonas. So the correct sequence is A, papaya, that is ring spot virus 2, B, BT cotton, 4 insects, C, Roundup Ready soybean, 1 herbicides, D, tomato, 5 Pseudomonas. So the correct sequence is 2, 4, 1 and 5. Question number 17, the correct answer is 4. Question number 18, assertion. The bruise-resistant transgenic tomato is developed by rDNA technology. Reason, it offers longer shelf life. Options 1, both A and R are true and R is the correct explanation of A. 2, both A and R are true but R is not the correct explanation of A. 3, A is true but R is false. 4, A is false but R is true. Okay, here assertion statement, as already we have studied that in tomato, the flavor savor variety of tomato had been produced which is having the bruise resistant property. What is this bruise resistant? We know that when the tomato plants are transported from the place of their production to the market. So what happens if the transportation is delayed? So these tomato plants are spoiled. So that ability is called as bruise disease, or the property is called as bruise disease. And if the process is delayed, the maturation is delayed, we call this as a bruise resistant property. So because of this rDNA technology, this bruise resistant property have been introduced into tomato. That variety of tomato is called as a flavor savor variety. That is correct. What is given the reason is, and we know that here when the transportation time is delayed, so the freshness of the tomato fruits is retained. That is nothing but the shelf life of the plants is increased. So they have the longer shelf life. So both A and R are correct and R is the correct explanation of A. So question number 18, the correct answer is 1. Question number 19, assertion. The rDNA technological process have immense impact in the area of healthcare. Reason, commercial chemicals are produced at lower costs by exploiting microorganisms. Options 1, both A and R are true and R is the correct explanation of A. 2, both A and R are true but R is not the correct explanation of A. 3, A is true but R is false. 4, A is false but R is true. Okay, here it is related to the health care. As we know that by using this rDNA technology, so we can improve the health of human beings. So that is how it is done means, yes, it is done by a process like let's take some examples. Human insulin is produced one thing, or the chemicals or the drugs, or we can say that pharmaceutical enhancement or development can be done. That is by using less amount we can get more number of pharmaceutical products. That is a way at low cost. By using low cost we can get more number of products, and that is done by exploiting the characteristics which are present in the microorganisms. So that both A and R are correct and R is the correct explanation of A. Question number 19, the correct answer is 1. Question number 20. Which of the following options can be used for increasing the food production? 1, waste treatment. 2, organic agriculture. 3, bioremediation. 4, genetically engineered crop based agriculture. Options 1, 1 and 2, 2, 1 and 3, 3, 2 and 4, and 4, 3 and 4. So here whatever the 4 that have been given are related to genetic engineering only, but the question is related to the food production, increase in the food production. So first one, waste treatment, it is not related to the food production, that is reducing the pollution by using various microorganisms. So first one is incorrect. Then organic agriculture, yes, here organic agriculture means by enhancing the various usage of various biofertilizers or chemical fertilizers, we can get the more production of the agricultural crops. So second one is correct. Third one is bioremediation, that is controlling the pollution by using microorganisms. That is also not related with the enhancement of the food production. And fourth one is genetically engineered crop based agriculture. We know that GMCs are produced, that is genetically modified crops are produced, which will give more yield and also have resistance to the diseases. So that from the given options 2 and 4 are related to the increase in the food production. So question number 20, the correct answer is 3. Question number 21. Microorganisms are commercially not extensively exploited for the production of which of the following compounds? 1. Vitamins, 2. Test tube plants, 3. Interferons, and 4. Vaccines. Options: first one 1 and 2, second one 2 and 3, third one 1 and 3, fourth one 3 and 4. Okay, we know that microorganisms are very much used in the production of various types of economically important plants or various components. So from that, here the question is not extensively used for exploitation. So those are, one is vitamins and second one is the test tube plants. But here we know that interferons or vaccines are produced on large scale by exploiting the properties which are present in the microorganisms. So that is in the process called as molecular forming. So from the given options, 1 and 2 are not related to this aspect. Question number 21, the correct answer is 1. Question number 22, the GM plants are useful in 1. Making crops more tolerant to antibiotic stress, 2. Developing pest resistant crops, 3. Enhancing nutritional value, 4. Increasing the efficiency of mineral usage. Options: first one 1, 2, 3 and 4, second one 2, 3 and 4, third one 3 and 4, fourth one 1, 2 and 4. So here it is related to the GM plants, that is genetically modified plants. So we know that GM plants like basmati variety, which is having the resistance to biotic and abiotic stresses, so that tolerance or tolerant to abiotic stresses. So this is correct. The best example where we can quote here, it is a basmati variety of rice. Then the second one is the developing pest resistant crops. So already we have studied that BT cotton like plants are produced which are having the resistance to the insects or pests. So that Bacillus thuringiensis have been used. So here we can say that the pest resistant crops are also being produced by this genetic engineering, that is GM plants have that property. So the third one is enhancing nutritional value. So here enhancing nutritional value, like we can say that a golden rice variety of plants are produced. So here that is produced by incorporating the genes which are responsible for increase in the vitamin A in the golden rice variety. So that nutrition enhancement of the nutritional value is also related with the GM plants. So then fourth one, increase in the efficiency of mineral usage. Of course here the plants will utilize the minerals which are present in the soil, but up to which efficiency of usage is also very important. That is, up to which level the plants are able to absorb all the nutrients which are available in the soil. So if GM plants have the ability to absorb large amount of minerals which are present in the soil, so that is also efficiency in nutritional usage. There is also important character related to GM plants. So from the given options 1, 2, 3, 4, all the 4 are correct. Question number 22, the correct answer is 1. Question number 23, biotechnology is concerned with 1. Utilizing the properties of microbes, 2. Exploiting cells and cell constituents, 3. Maintaining the parental characters in next generation, 4. Generation of useful products for human welfare. Okay, options 1, 1, 3 and 4, 2, 3, 4 and 2, 3, 1, 2 and 4, fourth one 2, 3 and 4. So from the 4 characters related to biotechnology that have been given in the question. So if you see the third option, maintaining the parental characters in the next generation, it is not related to biotechnology. Why? Because that is a property of heredity or inheritance, where the parental characters are retained in the next generation. So third one is incorrect. And according to biotechnology definition, we know that the utilization of the properties of the microorganisms are the characters which are present in the cells of the microorganisms for producing the disease resistant varieties or high yielding varieties are useful for the development and human welfare. So that utilizing the properties of microbes is correct, exploiting the cells and the cell constituents is also correct, and generation of useful products for human welfare is also correct. Question number 23, the correct answer is 3. Question number 24, study the following table. 1. Brassica napus, male sterile plants, reduce the cost of hybrid seed production. 2. Transgenic tomato, bruise resistant, herbicide tolerant. 3. Super weeds, gene pollution, resistant to weedicides. 4. Type A, improved nutritional value, rich in minerals. The correct combination is options 1, 1 and 4, 2, 1 and 3, 3, 2 and 3, 4, 1 and 2. Okay, here let us see the 3 combinations that have been given here. So first one is Brassica napus. Already we have studied that the male sterile plants are produced by Brassica napus. The male sterile plants are produced. So what is the importance of these male sterile plants? So here there is no need of emasculation, that is one thing, and also we can reduce the hybrid seed production, cost of hybrid seed production. So cost of hybrid seed production is reduced. So this is all the 3 combinations are correct related to 1. Then when it is coming to 2, so transgenic tomato. So transgenic tomato is a one which is having the property that is bruise resistant property, but when it is considered with the bruise resistant property, that is a transgenic tomato which is a flavor saver variety, but in the same way it is given as a herbicide tolerant, but it is not the one more tomato varieties transgenic tomatoes produced which is having resistance to pseudomonas but not to herbicide. So that herbicide resistant is incorrect. This one is incorrect. Then third one, super weeds. We know that when the genes are introduced into an organism, so in order to improve the quality and quantity of the plant, but unfortunately because of that gene manipulation, some new genes that is the actual sequence of the genes is changed, that may lead to formation of a super weed. So when it is forming into super weed, we call that as a improvement of gene pollution, improvement in the gene pollution. So here they will be useful for resistance to weedicides. So when it is done, resistance to weedicides. Then fourth one, type A variety. Type A variety is a rice variety into which vitamin A is or we can say genes responsible for vitamin A synthesis are introduced, so that nutritional value is increased, and that variety of rice is called as a golden rice variety. But here it is not the rich in, it is given as rich in minerals, but the rich in minerals is incorrect. In place of this one it should be vitamin A. So that from the given options 2 and 4 are incorrect, whereas 1 and 3 are correct. So question number 24, the correct combinations are 1 and 3. Question number 24, the correct answer is 2. Question number 25, study the following table. 1. DNA prevents expression of gene. 2. Insects Bt toxin active form. 3. Bt toxin gene cry. 4. Plants biofactor is molecular seizures. The correct combination is options 1, 3 and 4, 2, 1 and 3, 3, 2 and 3, 4, 2 and 4. Okay, here in the first one if you see it is given as DNA prevents expression of gene. So all these 3 are nowhere related to each other. So that first option is incorrect. Then coming to the second one, that is insects Bt toxin and active form. Already we have studied that the protoxins which are in inactive form, they are converted into active form. That is the toxins are produced, that is called as Bt toxins inside the insect, that will lead to the death of the insect. So this one is correct. Then the third one is Bt toxins, gene and cry. So Bt toxins are specified by some genes, and those genes are symbolized or written in the form of cry genes. So this is correct. Then fourth one is the plants, biofactories and molecular seizures. So here not the plants, but here we are using okay, here the plants are used as biofactories, but that is called as molecular farming, but not molecular seizures. Molecular seizures means the restriction enzymes which are useful for cutting the DNA molecules. But here plants and biofactories are correct, but it should be molecular farming instead of molecular seizures. So that is incorrect. So from the given 4 combinations, 2 and 3 are correct. Question number 25, the correct answer is 3.